I'm Dehan from City University of Hong Kong, and thanks for this opportunity to share our work, a tool for identifying the viral reassortment of segmented viruses. So compared to other viruses with single contiguous genomes like the SARS coronavirus 2, the segmented virus is very special because its genome is fragmented. For example, the influenza A viruses have separated fragments. And because of such a very special genome structure, the segmented virus can produce the novel virus string via a process called reassortment. So when multiple different virus strings infect the same host cell, their fragments will be mixed and then packed into the new viral particle. So the new viral particle may contain the fragments from different parental strings, and this may change the this new viral string, the properties, and sometimes the property may be harmful to the human. So this is not good news. And there are several influenza flu pandemic was caused by the segmented virus. And for example, in 2009, there is a novel reassortant strain, and this novel reassortant strain caused the 2009 flu pandemic. So because of the influenza continue to evolve and reassort, it is very important to be able to monitor and identify the reassortment event to help us to prevent the potential pandemic. So to identify the reassortant string, using phylogenetic tree is a good way because the reassortant string will contain the fragments from different parental string. So when we're checking their segment tree respectively, we will see the mismatch in the evolution relationship between other strings. For example, this string 4, we found the mismatch between these two segment tree because these two segments are from different parental string. So we can utilize such mismatch between the phylogenetic tree to find out the reassortment. And with the rapid development of the sequencing technology, we can obtain the genome sequences of the segmented viruses, and then we can construct the phylogenetic tree for each segment respectively. And then we can do a pairwise comparison between the tree to find out the discordance between the tree, and so that we can find out the possible reassortant strings. While this method is good, but it has some challenges. First, the phylogenetic tree contains the noises. That means even though the string is not a reassortant string, but its location across different phylogenetic traits will be different. So this may be misleading. And also when the tree gets larger, it is hard to compare the large tree and it will be computationally expensive and will be slow. So to address these challenges, we developed a tool called VReassort. And this tool will first construct the phylogenetic tree from the two set of genome sequences, and then it will extract the feature from the phylogenetic tree for each string. And this feature will include many information from the trees, like the branches, the internal nodes, and the subtree, so that we can utilize such feature to predict the reassortment. And then this feature will go through a Siamese neural network, and this neural network will learn how different this feature of different segment trees, and it will output a score to indicate if these two features are different. And to achieve the prediction, we need the data to train the model. However, there are limited data with no reassortment annotation in the current database. So we simulate the reassortment process and obtain the simulated data. and then we use such simulated data to train this model to predict the reassortment between the pairwise comparisons. So once we got the pairwise comparison between many trees, we can actually do the segment clustering to help us understand which segment came from the same parental string in the reassortment. For example, I used two different colors to represent their the origin of the segment, and we can, based on the pairwise prediction, to determine the cluster. And we also provided a module, the data quality evaluation, to help user to assess the reliability of the reassortment inference. So this is how our tool work for the viral reassortment identification. And let me show you some results. So we did many experiments to test the performance under various scenarios like here. And here I will show one example. This is the example of the simulated data. And we can see our tool with the red curves achieved the highest F1 score. And when the data get larger, like about 1,000 string in the data set, and we can see our tool can finish about 1.5 minutes. So this is quite fast. And in the following slide, I will introduce the application of our tool to find out some interesting pattern on the real influenza A virus string between the 2010 and 2022. So here is the example, and among over 7,000 influenza string, our tool identified 2,000 something reassortant string. And within this string, if we check if their host, we found that the swine string has the highest reassortant proportion as shown in this orange curve. And also we can see from this figure that the reassortant proportion across the year is fairly stable. So, and when we check the subtype of this reassortant string, we will see a very special case is the avian string. The avian string with different subtype will show very different reassortant ratio pattern. For example, here we can see they have different ratio, and for these two subtype with high ratio, we can see these two subtype string can also infect the human. So we need to pay some attention to such subtype to monitor the reassortment event for them. And besides the host and the subtype for the reassortment, I also investigate the reassortment pattern on the segment level. And it is not surprising that most of reassortant string originate from only two parental string. As shown in this figure, different color represent the different parental string sources, and we can see there are many string with only two parental string. And when we look into more detail between these segment exchange, we will see the exchange rate between pairwise segment, and we will see some segment have the low exchange rate. And when we check such exchange rate, we found that these segments are functional independent. For example, for the HA and NA segment, they have much lower exchange rate than other segment pairs. So this is maybe because the HA and NA must stay balanced when they do the receptor binding. So our finding align with the current result and the findings. So this is the case to show how to use our tool to identify the reassortment and do some reassortment analysis. So thank you for your attention. And our paper was published in Nucleic Acid Research. You can find more detail on this paper in this paper. And this is our tool. You can scan this QR code to obtain the tool and use it if you are interested. And our group, Professor Yan Nisheng, we developed many viral tools for the viral analysis, and you can visit our website if you are interested. And thank you.